Hello everyone, and welcome to this week's EV Fundamental Biology series talk. My name is Helena Branco. I'm a PhD student at both the University of Porto in Portugal and University of Torino in Italy. And together with my colleague Drishya Mainali from the University of Sydney, Australia, I'll be moderating this talk. Today it is my pleasure to introduce our speaker, Professor Edith Buzás from the Semmelweis University in Budapest, Hungary. As many of you know, Professor Buzás is a former ISER president, co-organizer of the 2024 Gordon Research Conference on EVs, of the 2025 ISER Biomolecular Corona Workshop, and is a true pioneer within the EV field. In this session, she will uncover how absorbed biomolecular coronas can mask extracellular vesicles, hindering their detection and identification. She will also discuss how smaller EVs can be released within larger vesicles and how EVs can remain hidden in the extracellular matrix or cellular debris, challenging our understanding of EV visibility and characterization. Before we start this session, I would like also to remind you all to please post your questions in the chat box during the presentation, or otherwise to feel free to unmute yourselves and ask them yourselves during our open Q&A session at the end of the talk. Now, I welcome Professor Edith Buzás to start her presentation and thank her for accepting our invitation.
Thank you very much, Helena, and thank you all for kindly inviting me to this seminar session. It's my pleasure, and I'm very honored to be with. Remove from the screen the photo to the bottom so that it wouldn't interfere much with the image. So I give the title to the talk, EVs with Mass Identity. And what I would like to discuss with you and briefly overview with you is what factors can interfere with the identification of EVs. I would start with the lack of certain EV markers, discuss briefly the inside-out EVs. As you heard, I would like to touch upon the EV biomolecular corona. I would like to discuss EV mimicry, enzymatic modification of EVs, extracellular matrix interactions, storage-related masking effects, and the presence of previously ignored and not identified EVs. Let's start with the first topic, the lack of certain EV markers. I think this is something that you are all familiar with. Even MySEF 2023 emphasizes that there is no universal EV marker, and many EVs will not be detected if our assays rely on a single canonical marker, like a tetraspanin marker. And I would like to just remind you that in 2016, a PNAS paper from Clotilde Thierry's lab already provided evidence that if we use pull-down experiments with anti-CD81, anti-CD63, or CD9 antibodies, we are able to separate, identify. A more recent paper, again from Clotilde Thierry's laboratory from 2021, demonstrated that exosomes versus small ectosomes could be shown when these vesicles are released from a single cell line, and we can distinguish small EVs which are enriched in CD63 from those which are enriched in CD9, and even we have a subset which shares both surface tetraspanins. I would also mention a paper from Randy Carney's laboratory where he pointed out that tetraspanins are unevenly distributed across single extracellular vesicles and biosensitivity to multiplex cancer biomarkers. If we capture EVs with a specific antibody, and then we use a detection antibody that would detect a tumor biomarker, we detect less than if we use a non-specific EV capture and then apply this tumor biomarker-specific detection antibody. So said that, clearly we have to be cautious. We should not expect to find all three tetraspanins on our vesicle preparations. We may have mixed population in our prep. We may have the presence of all three tetraspanins, but it very well can happen that we only detect one or two of them. The second topic I would like to discuss is the possibility and presence of inside-out vesicles. As you know, when vesicles rupture, they might release their inner content. However, because it's energetically more favorable, they close back to a vesicle format again. However, it may happen that under certain circumstances, this reclosure occurs in an inside-out way, and the inner phospholipid bilayer will be on the surface, and the outer phospholipid layer of the bilayer the outer leaflet will be inside the vesicles. Now, we do know that vesicle damaging conditions include forced filtration, when we try to size filter our vesicles, or when we subject them to centrifugation, especially when we subject them to very high centrifugation forces. Most probably, very often we encounter this phenomenon that our vesicles open up and close back again. And theoretically, it's possible that sometimes the vesicles close back in a reciprocal form. The evidence from biology is quite abundantly available for such inside-out vesicle formation. I would like to just quote two articles. Both describe red blood cell membranes. And they describe that when red blood cell membranes are exposed first to hypoosmotic, then to normoosmotic or hyperosmotic conditions, they typically seal back in an inside-out membrane vesicle form. And also, when hypoosmotic lysis of red blood cells occurs, and then we incubate the red blood cells at 37 degrees centigrade for 20 minutes, there will be a mixture of inside out and outside in vesicles in the preparation. So it's theoretically feasible. It may happen in our extracellular vesicle preparations as well. And indeed, if you have a look at the cross sections, ultra thin cross sections of large size extracellular vesicles, you can see that the pellets, even a low-speed pellet, a 12.5 K pellet, can contain damaged vesicles with ruptured membrane. Typically, this membrane rupture and closure larger vesicles, it's more probable to occur in large-size vesicle populations. However, we cannot rule out that it happens also to small EVs. And I would like to mention this paper of Jan Lutwer's laboratory, where they compared by mass spectrometry the proteins in simple EVs, EVs treated with proteinase K, and EVs treated with trypsin, lysin C protease. And as you can tell, the biotinylated protein carrying vesicles also included some which were in unconventional orientation, like the SCAM3 or syntaxin4, which raised the possibility that indeed inside-out vesicle formation is there. However, I would say that it's probably not a very major, not a very frequent phenomenon. The next point I would like to talk to you about is the formation of the biomolecule corona. In 2016, we found that when we incubated commercial LDL lipoprotein particles stained with osmium tetroxide with nascent medium-sized extracellular vesicles, the mixture immediately showed very interesting corona formation on the surface of the nascent EVs. These nascent EVs were freshly released into a cell-free conditioned medium. As you see, the lipoprotein particles, which are highly electron dense, are clearly forming a corona around the periphery of the vesicles. And we could confirm the simultaneous presence of both CD81 and ApoB around these vesicles in our preparations. So this was an evidence that lipoproteins can actually associate with the surface of EVs and form a corona. The other interesting observation came 1 year later, where we found that in a cell line that was exposed to an antibiotic treatment, ciprofloxacin treatment, that is known to induce genotoxic stress in mitochondria. When we looked at the density gradient fractions of the vesicles secreted into the medium, what we found was that we found CD63 positive events in 2 fractions. However, only in 1 fraction, in fraction 7, we found something very unexpected. We stained these vesicles also with propylium iodide, the DNA dye, and as you can see, this positivity is not showing up anywhere else, just in fraction 7. In the fraction which contains CD63 positive vesicles, but this was the one with higher density. In additional experiments, we could show that in these fractions, the vesicles were indeed coated with DNA. This was predominantly mitochondrial DNA, and this was susceptible to DNase 1 treatment. The last question we asked was, could it be that there is a protein association with the surface of EVs to form a protein corona around the vesicles? In the synthetic nanoparticle field, it's easy. They immerse vesicles or synthetic nanoparticles, sorry, not vesicles, synthetic nanoparticles in biological samples, and whatever is adsorbed onto the surface is evidently corona. In the case of vesicles, both the vesicular proteins and the potential corona proteins belong to the same species. So it was not so evident how to distinguish them from one another. Anyway, we took this approach to isolate nascent THP1 cell-derived vesicles. Again, nascent means freshly released into serum-free medium. We also tested washed platelet large EVs, and then we incubated them in EV-depleted blood plasma samples. We took 18 healthy subjects, 16 RA rheumatoid arthritis patients, and the individual EV-depleted blood plasma samples, we immersed these nascent EVs, incubated for 30 minutes at room temperature, and then afterwards, we re-isolated them with differential centrifugation, ultracentrifugation, density gradient, ultracentrifugation, or size exclusion chromatography. And then we subjected them to analysis by mass spectrometry. We considered that if we take the complete protein list and we subtract the proteins which were only present in the nascent EVs, the difference will give us the corona components. Around this central EV, you can see the potential corona proteins that we could identify around our extracellular vesicles. And these little icons demonstrate to you whether we could recover these proteins after different reseparation methods. As you can see, certain proteins could be recovered by all 3 reseparation methods. However, there were some others which were only present, for example, in the differential centrifugation preparation. Anyhow, we compared our proteomic hits with those that have been published for viruses: respiratory syncytial virus corona, herpes simplex virus corona. And again, from the literature, we took proteomic composition of the positively or negatively charged lipid nanoparticle coronas. And we compare these published corona proteins with those that we identified around the vesicles, and we could identify 9 shared corona components. These included, as you can see, ApoA1, ApoB, ApoC3, ApoE, complement 3, 4, fibrinogen, and immunoglobulin heavy chain molecules. I add albumin also. This is a little bit separately indicated because we consider that corona proteins are those which are adsorbed onto the surface of the vesicles and not expressed by the EV releasing cell. Now, albumin was an exception to the rule because albumin was also expressed by the EV secreting THP1 protein, and it's a major component of the EV protein corona. When we looked at the deposition of the different corona proteins around the green fluorescent vesicular surface, we could see that certain proteins, corona components, formed a patchy, more or less diffuse corona, whereas in some cases we saw individual large aggregates of proteins attached to the surface of the vesicles. So we consider that in biological systems, as soon as vesicles are released to the extracellular environment, extracellular proteins are necessarily adsorbed onto the surface of the vesicles. No matter whether these vesicles are ectosomes shed from the plasma membrane or these are vesicles released from the endosomal compartment. And importantly, certain proteins are attached to the vesicle surface already inside the cell. If the vesicle enters another biological environment, it is presumed that there is an affinity-based competition for the binding to the EV surface, and higher affinity binders replace lower affinity binders. The major question here is that could it be that this EV corona indeed mask EV surface? Could it be that, for example, antibody recognition is interfered with the corona proteins? Could it be that antibodies recognize EV surface molecules better if the corona is absent? To address this question, we took this approach that I'm showing you in this figure. We again tested green fluorescent EVs. These are from the HEC293 palm GFP cells, which have been originally generated by Charles Lai. And these green fluorescent, again, nascent EVs, released into serum-free medium, they were co-incubated with normal human plasma proteins that we labeled with Cy5. And what we saw is very similar to what we found when we co-incubated the vesicles with lipoproteins. As you can see, there is clearly a corona formation around the surface of the green fluorescent vesicles. We tried to remove this corona by different procedures. We subjected the vesicles to size exclusion chromatography. However, in our hands, size exclusion chromatography did not make a difference. It did not remove the corona efficiently. The same was the case when we exposed the vesicles to twin. Twin did not remove the corona. The corona did not detach in the presence of beta-mercaptoethanol, and EDTA was also not able to detach the corona. However, when we exposed the vesicles to the high ionic strength, 1.5 molar sodium chloride, lithium chloride, or potassium chloride solution, as you can see, a very significant proportion of the corona components detached from the EV surface. Our original question was, if you remember, that whether this corona can interfere with antibody recognition, immune recognition of the vesicle surface molecules. So what we did was we used the MaxFlex kit, and we compared the detection of certain surface molecules. There are 37 in the MaxPlex kit, on small EVs from blood plasma in the absence or presence of high salt washing. And as you can see, almost half of the detected surface markers showed an improved detection, an enhanced positivity in the presence or in the case of high salt washing prior to detection, which told us that indeed the corona might interfere with antibody detection of the vesicles. We also asked the question, could it be that this corona interferes with the annexin-5 phosphatidylserine interaction on the outer membrane leaflet of vesicles? To address this question, we immunocaptured certain vesicles by magnetic beads that carried annexin-5. And as you can see, we found a significantly increased immunocapture when the corona was removed by high salt washing by using a prior 1.5 molar sodium chloride wash of the EV preparation. And when we used fluorescent labeled annexin-5, again, we found the same phenomenon. We found an improved detection of the vesicle, an improved interaction of the annexin-5 and phosphatidylserine if we carried out a prior high salt washing. The next point I would like to talk to you about is EV mimicry. And EV mimicry is important because we now know that there are so many different non-vesicular extracellular particles. In this figure, you see these include, for example, lipoprotein particles, nucleosome ribonucleoprotein particles, exomeres, supermeres, release granules, virus particles, protein aggregates and complexes, crystals, eosinophil and neutrophil released extracellular traps, calciprotein complexes, microplastics, and more. Let's have a look at the mimicry by lipoproteins. As you know, lipoproteins include HDL, high-density lipoprotein, low-density lipoprotein, lipA, which is recognized as a very important lipoprotein, the detection of which is important in the diagnostics of cardiovascular diseases, IDL, chylomicron remnants, and VLDL particles. And importantly, these lipoprotein particles can aggregate, they can form hybrid particles with EVs, and they are components of the EV corona, as I already showed you before, demonstrating it on an electron microscopic image. Now, let's talk about HDL first. It has been demonstrated that HDL can clearly interfere with the detection of extracellular vesicles because HDL has the same flotation density as EVs. And this is important because otherwise the diameter of HDL particles is substantially lower than that of the EVs. So it is just 7 to 13 nanometers in diameter. However, because of the shared flotation density, it might be misleading when detecting EVs. And when we look at the LDL particles, they are also quite tiny, 18 to 23 nanometers in size. So you cause much interference, but it's not the case because LDL particles can aggregate. Phospholipolysis, oxidation, and proteolysis can cause aggregation of the LDL particles. And in this case, when there is LDL particle aggregation, they immediately gain the size of EVs, and obviously they can be interfering with vesicles during detection. Another mimicry is caused by protein aggregates and complexes. And I'm just showing you 3 electron microphotographs demonstrating pellets with EVs from synovial fluid samples of patients with rheumatoid arthritis and osteoarthritis. As you can tell, indeed, there are EVs in the pellet, but almost in the same size range, we have clearly protein aggregates or protein complexes. And indeed, what we found was that in these autoimmune diseases, the protein aggregates and immune complexes shared size distribution with EVs, shared light scattering properties, and shared sediment atomic force microscopic images of EVs and these protein complexes. They looked quite alike. NTA did not really distinguish them from one another. And when doing flow cytometry, simply based on flow forward scatter, side scatter, as you can see, the immune complexes or protein aggregates gave very clear EV mimicking signals, for example, in the case of OVA and anti-OVA, IgM, anti-IgM. Importantly, such protein complexes are not only immune complexes formed by antibodies and antigens, but also we have to add that avidin biotin complexes can be also giving the same interference. If you add Triton detergent, as you know, the vesicular structure membranes are sensitive to Triton lysis, like to 0.1% Triton X100, whereas the immune complexes and other protein aggregates would be resistant. So this is a simple way to distinguish EVs from non-vesicular extracellular particles, because non-vesicular extracellular particles might be resistant to detergent lysis. If you take the total event number, take away the remaining non-susceptible population, and you refer to reference beads, then you might get the actual EV number. I have to add that we are currently working on refining the detergent lysis technique, because with the newer, higher resolution flow cytometers, we may have to redefine particular conditions. Some of these new flow cytometers will be able to also detect detergent-based micelles, and therefore there is a little bit of need to further improve the detergent lysis method. I'm just showing to you one more example. If you apply anti-IgM or anti-IgG staining to plasma samples of controls and SLE patients, and you apply the Triton lysis, you see in SLE there are really large numbers of particles which are resistant to the detergent lysis, meaning that they are not vesicles, they are probably antibody-antigen complexes. I would like to also drive your attention to protein aggregates, which are present everywhere. In this particular case, you see that the particle diameter looks quite clearly matching the expected one, both before and after Triton lysis. There is not much change. However, if you compare it with the real vesicle preparation, you can tell that vesicle numbers drop after detergent lysis. You can recognize these protein aggregates in samples by immunogold electron microscopy. And one interesting finding that I would like to drive your attention to is that repeated centrifugation may induce repeated formation of protein aggregates. In this particular experiment, we used 2 times 2.5 thousand G centrifugation for 15 minutes to produce platelet-free plasma. Then the sample was filtered through a 5 micrometer pore size filter, then subjected to a 0.8 micrometer size filter. Then we applied 20,000 G and 100,000 G. And then after all these centrifugation steps, we had the supernatant, which we consider that could be an EV depleted plasma sample. Now, this EV depleted plasma sample was subjected to this low speed centrifugation once, twice, 3 times, 4 times, 5 times, 6 times. And have a look. Even after 6 times, we still had a distinguished palette. which suggested to us that we continuously produce aggregates. In this pellet, there was no lipid detectable. However, there was protein, even though its amount was decreasing. And when we compared these to extracellular vesicles, both EV protein and lipid content was comparable all the time. Another mimicry can be caused by release organelles, for example, mitochondria or release granules of granulocytes. In this left-hand side electron microscopic image, you see the pellet of a microglial cell line, BV2 cell line, and again a 12.5 K pellet, which was produced after 60 minutes centrifugation. What you can see is that you have multilamellar bodies in the pellet and also clearly mitochondria. At least 3 mitochondria can be recognized in this single pellet. When talking about granulocytes, mast cells, eosinophils, or basophils, and neutrophils, the granule release is phenomenon. However, it has not been properly dissected yet, whether the release particles are granules, mixture of granules and vesicles, or maybe vesicles themselves. So this opens up a very interesting and exciting new field to explore. We always have to remember the mimicry by viruses, and please allow me to mention a personal example. When we, with my colleagues, started to work on extracellular vesicles, we used this BV2 microglial cell line. We applied the protocol 100,000 G pelleting, looked at the pellet with a transmission electron microscope. Please note that this is not the regular preparation that you often see. It's not that a drop of EV suspension has been placed on a grid. We rather took the pellet and made cross-sections, ultra-thin cross-sections of the pellet. Well, there were some larger size vesicles, but we were super happy seeing how many beautiful, identical looking exosomes we had. This was probably one of our first EV preparations. But then we had to sadly recognize that these structures were not exosomes. They were not small EVs. They were actually virus particles because these BV2 microglial cells were transferred by recombinant retrovirus. One has to be very cautious about viral mimicry in all EV preparations. Mimicry by nanoplastics is an emerging topic. Importantly, our whole environment is loaded with plastic disposable items. And even if you say that you are not using these flasks for drinking water, therefore nanoplastics is not a relevant topic to you, maybe you are a little bit mistaken, because plastic tubes may release nanoplastics into the solutions that you are using. And as you can tell, the nanoplastics could give very similar size distribution that what you find in the case of your vesicles, maybe an even more pretty homogeneous population of particles you can detect. And not only NTA can be used to identify these nanoplastic particles, but also flow cytometry settings that you apply for vesicles could be quite applicable also for these nanoplastic particles. They most probably contribute to the noise that you experience in your samples. Enzymatic modification of EV markers is another topic. Importantly, if proteases or glycosidase enzymes are present, they can cleave or shed surface molecules from extracellular vesicles, and they can actually remove them or simply modify them, truncate these surface molecules, or can simply alter their glycosylation. I would like to remind you this paper from last year in which brain-derived extracellular vesicles have been isolated from brain tissue. These days, it's a very important point to isolate vesicles or separate vesicles not only from biological fluids but from tissues. And also from brain tissue, it is an important experimental approach. As you know, the original approach is to separate extracellular vesicles from tissues by applying collagenase or other proteolytic enzymes to release the vesicles. However, authors of this paper have clearly demonstrated that even minor enzymatic digestion can induce artificial proteolytic processing of the brain-derived extracellular vesicle markers such as flotilin or CD81, and only the enzyme-free isolation was the one that really preserved EV surface molecules in their hands. The next point is masking by extracellular matrix components. And I would like to refer to the matrix-bound vesicles. This is important, particularly if you are looking at vesicles which are secreted by matrix-producing cells. So if you seed your cells to a tissue culture plate, the cells produce extracellular matrix, and then you harvest conditioned medium from this culture, you might completely miss those vesicles which are released to the matrix and which are stuck in the matrix. These vesicles are referred to as matrix-bound vesicles. And what you can see is that they represent a completely different, distinct population of EVs. In terms of RNA, they definitely seem to be different from liquid phase EVs and also from the releasing cells. And their lipid content also distinguishes the liquid phase EVs from the matrix-bound vesicular EVs. Storage might be a masking factor. First of all, you probably also experience adsorption to tube walls. Therefore, you may miss your vesicles just because they are adherent to your tube wall. Importantly, during storage, degradation of markers can occur, decomposition of fragments, decomposition of EVs into fragments, and they can also aggregate or fuse. And this is just one simple example that, for example, red blood cell surface marker CD44 during red blood cell storage at low temperature, but it may occur to many surface markers. Finally, I would like to drive your attention to the point of previously ignored populations of EVs. Many of our EVs, I suggest that are hidden in what people called debris. I find this word debris rather harmful because we sweep everything under the carpet. We don't bother looking into this debris. Therefore, for decades, we only focused on small EVs, and this debris was very often discarded. I call by debris the low-speed pellets that we obtain, for example, from conditioned medium. And the components were neglected for a very long time. Nowadays, we come to the understanding that this debris is not a real debris. It's a collection of very large size vesicles. And a completely new field is emerging that focuses on this debris or very large EVs. Lastly, let me just drive your attention to a previously ignored population of EVs, which we could not observe until we carried out in situ fixation and analysis. Let me explain to you what I mean by this. In situ fixation meant that we were growing our cells on gelatin fibronectin coated surfaces, and then removed the vast majority of the tissue culture medium and added the fixative right into the thin layer of medium on top of the cells. So without pipetting, without resuspending, without centrifuging, without touching, we fixed them in situ. And look what we have found. We tested HEC-PALM GFP cells, regular HEC293 cells, HEPG2 cells, HT29 cells, HL1 cells, and bone marrow-derived mast cells as primary cells. And in all instances, we could observe in these in situ fixed cultures the protrusion of multivesicular large vesicles from the surface of the cells. We also could observe detached large vesicles, multi-compartment vesicles, in all instances in the vicinity of the cells. There was already no connection with the releasing cell, and within one limiting membrane, we found many little vesicles. And finally, we also found evidence in all instances for disrupted membrane of these large vesicles. We refer to this phenomenon as a Thornbeck release of internal small vesicles. We found it in all instances. We could also observe the same thing in ultra-thin sections of different organs from, this is from mouse kidney, you can see different examples, or mouse liver. We have done a lot of electron microscopic analysis of different other organs of the mice and also from human tissues. And I can tell you that this phenomenon is universal. So there is a large size vesicle that has not been seen before because the membrane around this vesicle is not very stable. Within a couple of minutes, we see a spontaneous rupture of this membrane. Therefore, when we pipette our samples, centrifuge our samples, we will miss these large bags. We will only see the smaller inner vesicles. When we looked at the different markers on the small inner vesicles, what we could see is that they were, as we expected, positive, for example, for CD63. But importantly, we could also observe an autophagy marker, LC3B, on the surface of the small inner vesicles. However, the co-localization experiments were very surprising. Small EV marker, ALIX and CD63, well co-localized. Same was true for ALIX, CD81, TSG101, CD63, TSG101, CD81. So the different small EV markers beautifully co-localized with each other, but never co-localized with the LC3B positive little vesicles. These are two additional cell lines, and in all instances, we found the same. Small EV markers beautifully co-localized. These are the markers that we are looking for in small EV preparations, but they did not show any co-localization with the LC3B. We found the same thing in electron microscopic images using immunogold technique in small EV preparations. The vesicles were either LC3B positive or CD63 positive, but never simultaneously positive for LC3B and CD63. We could use super resolution microscopy to identify in the cells multivesicular bodies which contain CD63 positive inner vesicles, autophagosomes which contained LC3B positive inner vesicles, and the fusion product of autophagosomes and MVBs that are referred to as amphisomes. And as you can see in these amphisomes, we find both LC3B positive vesicular structures and CD63 positive vesicular structures. This is also an ambisome. And finally, well, I'm not showing the details, but finally we have come across these possible mechanisms that we propose as a Thornbeck mechanism of small EV release. Multivesicular bodies have CD63 positive inner vesicles. Autophagosomes have an inner membrane that is known to be LC3 positive. And when the multivesicular bodies fuse with the autophagosomes, in this fusion product, in the amphysome, we observe the degradation, fragmentation of the LC3 positive inner autophagosomal membrane. What we propose that these fragmented membrane parts curl up to form inner small vesicles. And in a mature amphysome, we find the CD63 positive MVB-derived real small EVs. and the autophagosomal inner membrane-derived small EVs, which are LC3 positive. The cell will release these big structures by budding, by ectocytosis. The released large vesicles are the amphiectosomes, amphi because they come are released by ectocytosis, and ultimately there is a spontaneous release of the inner small vesicles, which are either CD63 positive or LC3 positive. And the reason why I'm showing this to you is because when we are looking at our small EVs, we are looking for the tetraspanin markers, but we forget to look, for example, for the LC3 positive small EVs, which look exactly the same, and they are in the same prep. Looked vesicle populations that have not been identified so far. I would like to add the last statement about this. This mechanism that we describe here seems to be the constitutive small EV release mechanism of resting steady state cells. When a cell is not stressed, not induced, this is the constitutive way of secretion of small EVs. So finally, I would like to sum up. We talked about the lack of certain EV markers, tetraspanins, the possibility of having inside-out EVs in our preparations, which is probably not a major component of missing EVs, the masking effect of the corona, a very significant EV mimicry with many non-vesicular extracellular particles, the possibility that we are experiencing enzymatic modification of the EV surface, and that's why we are missing certain EVs. Extracellular matrix interactions might hinder EVs. Storage can cause degradation, aggregation, fusion, or adherence to the tube wall of our vesicles. And there might be previously ignored vesicles which have not been identified yet, and we are not looking for them. That's why we are ignoring them. With that, I would like to thank the contribution of my team members, and thank you for your attention.
Thank you so much. That was fantastic, very comprehensive, as well as educational. So now please put any of the questions that you have in the chat, and I would like to go first, actually, while there are still some questions. My question is mostly related to the biomolecular corona part of the talk. I wanted to ask about how specific these protein corona is, especially in your nascent EV experiment. Do you think these EVs are in any way pre-primed for a specific corona at the hard corona level, or they're just a reflection of the biofluid they're in? So if you were to have those nascent EVs in different biofluids, at any point would they be just have like a pre-priming for specific interaction, or they're just plainly a reflection of the biofluid? What do you think?
Yeah, so I think most of the corona components are those universal ones which adsorb onto the surface, being abundant plasma proteins. However, in distinct locations, in distinct microenvironments, maybe this situation changes. And also depending on the releasing cell and the surface receptor composition of given vesicles, the receptor-bound ligands might modify the corona composition quite significantly. So it really depends on whether we are looking at any cells and a typical corona around them, or we are looking at specific cell-derived vesicles and their coronas. Obviously, if there is an inflamed body, there's an inflamed tissue, we can expect much more acute phase proteins, for example, adsorbed onto the surface of vesicles than in a healthy subject. So depending on the functional state of the organism, we can have different changes in the composition of the corona components.
Thank you. I will also address some of the questions in the chat. One's from Bhuvan. During corona removal from EVs, is it not affected by other surface markers of EVs, mainly in the case of using tween 20 and EDTA treated EVs?
I don't see this question, but it doesn't matter. If I understand, the question is whether if we use tween 20 or EDTA, whether the corona is not removed or not, or it is removed. We found no evidence for significant removal of the corona.
I see. And another one from Masha. Thank you for the nice presentation. Did you check the effect of different EV storage solution on biomolecular corona?
Did we check the effect of? Can you repeat it again, please?
Yeah, no worries. Did you check the effect of different storage solutions that you tried on the corona?
No, we haven't checked that.
And another one from Jonathan is when you refer to washing with sodium chloride for corona removal, what did you mean? Can we spike the total EV isolate with sodium chloride and then proceed to antibody separation?
So that's a very important question because if you try to reproduce this data, you have to be careful. Washing means in this case that you centrifuge your EV preparation in the presence of high salt concentration. It's probably the combination of the high ionic strength and also the shear force when you centrifuge your samples that ultimately results in the detachment of the corona. If you just soak your vesicles in a vial which has high salt concentration, by itself it's not sufficient to detach the corona. You have to centrifuge your sample either with low speed 12,000 K, 20,000 K, or 100,000 K in the presence of the high salt concentration.
Yes, I think Katerina's question's just similar as well. She asked if you think we should treat our EVs with sodium chloride prior to staining and measurement, for instance, analyzing them with flow, which
It's really up to you, but as I try to explain to you, you might get improved immunostaining if you do so. For sure enough, you have seeing is believing, so you have to try it. If it works for you as well, then well, I guess it's a good way to improve your detection.
yeah.
I also have to add those proteins that show the very much improved detection were listed on the slide, but the canonical tetraspanins have the same detection level, irrespectively whether we apply high salt washing or not. So if you are just looking at the tetraspanin positivity, maybe it's not necessary.
That makes sense. Jonathan just had a follow-up question about the treatment with sodium chloride if it would cause osmotic lysis.
No, we looked at the vesicles, and what we found was that in the presence of 1.5 molar sodium chloride, lithium chloride, potassium chloride, instead of undergoing an osmotic lysis, the vesicles lost water, so they became shrunken. They look like as raisins in an electron microscopic image. And if we normalize the osmotic conditions, then they regain their normal size and shape.
That makes sense. And Albert asked a couple. Have you performed digestion of the highly present glycans of the EV corona? Wouldn't these be these glycans the molecule that hinder the most accessibility to the surface of the EVs?
That's a very good point. No, we haven't tried it, but it makes sense. So I encourage you to try it because indeed you might have some improvement of the surface molecule detection by removing the glycans. We haven't done it.
And last question, sorry. Do these MBV-like EVs that you showed at the end of your talk have anything to do with the so-called microsomes?
No, no. First of all, many of the cells, in the case of which we demonstrated this constitutive Thornback release mechanism, many of them are non-migratory cells. So we don't have long retraction fibers, which is a hallmark for migrosomes. There's also a tetraspanin 4, that has been postulated to be a marker for migrosomes, and we don't have it. Actually, it's interesting. Some tetraspanin we might find inside our vesicles, but not on the outside, as in the case of migrosomes. But I agree, there is some similarity in terms that both migrosomes and these amphiectosomes have a pomegranate-like appearance with a limiting membrane and inside smaller inner vesicles.
Great. Masha asked another question. After treatment of EVs with twin or trident, did you check the lipid profile too, or just check the proteomic profile?
We only check the proteome. Yeah, we haven't looked at the lipid composition.
Okay, great. We're just on time. That was a fantastic discussion and loads of good questions. I would just like to thank Edith again for your time and everybody here that attended, and hope to see you all soon. But thank you very much. That was great.
Thank you so much for inviting me. It was a pleasure for me as well.
Thank you.
Thank you. Have a nice rest of the day. Bye.